And now I'd like to introduce Dr. Zeynep Kosaloglu-Yalcin back to the stage. So Zeynep was presenting the database and the query and reporting features, and she will now be talking us through the various cancer meta-analyses that we've conducted to date. So thank you very much, Zeynep.
Thank you, Nina. Let me share my screen. Yeah, so as Nina mentioned, our last talk is about the meta-analysis that we performed using CEDAR data. This is basically showing CEDAR in action and will demonstrate what you can do with the data. So when we first started building CEDAR, we wanted to test its utility on two very different types of cancer antigens, and we selected two specifically. On the one hand, we have the prostate cancer antigens. These are shared, non-mutated epitopes, and they are associated with prostate cancer, which is known to be a cold tumor. And we also have the neoepitopes, which are mutation-derived epitopes, usually from highly mutated and frequently mutated tumors, and these are often used in personalized cancer vaccines. So these two subsets, there are distinct and orthogonal categories because one, the prostate cancer category is rooted in decades of tumor immunology. It is very well studied, very well characterized. On the other hand, we have the neoepitopes. These are emerging targets. These are usually patient-specific, so very few are shared. And these are also usually harder to validate. So when we started to curate data, we created mainly these two categories. As it was mentioned in the earlier presentations, but now we are curating all the categories depending on the year they were published. But this is what we started with. So once we had enough data, we wanted to perform the meta-analysis with two main goals. First, we wanted to show and demonstrate the capabilities of CEDAR, and we also wanted to see if we need to improve anything to capture these very distinct categories of antigens. And also, of course, we wanted to identify any patterns or any gaps and see how the existing cancer epitope literature looks at the moment. So I'll start briefly with the prostate antigens analysis. This work has been published before. We'll also show the paper in a few slides. So these are just figures and tables out of the paper. So this first table, we can see a breakdown of the data we had in CEDAR for the prostate-associated antigens and epitopes. And as you can see, we have 8 prostate-associated antigens, namely PAP, PSA, PSMA, ROS, PSCA, STEAP, and TRP, P8, and TARP. So we selected, we curated these 8 antigen types, and we found a total of 278 epitopes that were curated from these antigens in a total of 132 publications. So the 3 most studied antigens are these first 3, PAP, PSA, and PSMA. You can see they dominate these numbers in terms of references, but also in terms of proteins, in terms of epitopes. So this is also where we'll go into a little more detail in the next few slides. So one interesting finding we saw here, we also showing the protein length here. So the number of epitopes did actually not correlate with the protein length, but it mostly correlated with the number of publications available for antigen. So we can already see there's kind of a study bias towards these proteins. And it's not dependent on the length of the protein, because one might argue, oh, these proteins are longer, that's why they have more epitopes, but that was not the case here. Next, we had a look specifically at the T-cell assays for the epitopes from the top antigens. As I mentioned, we're focusing on the top 4, and we wanted to see what kind of assays have been mostly reported for these. Overall, we captured nearly 1,400 assays across several assay classes, so including the T-cell assays, the tetramer staining, and the MHC ligand assays. And as we can see here, we also have the breakdown into class 1 and class 2 restricted epitopes. And we can see that class 1, here we have the total number, dominates the data. But we also have some class 2 epitopes, specifically from the PAP antigen. So this seems to be more, have more than the other antigens, some class 2 restricted epitopes as well. And what we can also see here that the tetramer data is quite sparse, and only a handful of epitopes have been validated with tetramers. Yeah, so this is highlights another knowledge gap in terms of assays that are being used to study these epitopes. Next, we look at the specific cytokines that were measured in these T-cell assays, and we can clearly see that interferon gamma dominates across all the antigens. So we have the highest numbers measuring the interferon gamma. We also have a few of the other cytokines like TNF-alpha, IL-10, IL-12, and so on. But these are only in the single digits, and we only have them for PSA, which is the most studied antigen. And for the other antigens, we only have like sparse data regarding these cytokines, which again highlights a study bias and a gap in our knowledge regarding these antigens. So just a quick conclusion for the prostate-specific antigens. This was a relatively small data set. So it was the first meta-analysis we conducted and also published. We found some important knowledge gaps. They can now inform the field, and hopefully people will be more motivated to analyze and to investigate the missing parts of these antigens and the different assays, and to have a more more granular view of these and what these antigens are doing. So the literature is highly skewed towards the top 3, PAP, PSA, and PSMA, and the other 5 antigens remain under-characterized. So this might be a good opportunity to invest some time to also investigate the other antigens. The T-cell assays are mainly restricted to class 1, and mainly the cytokines other than interferon gamma are not well studied. As I said, this has been published almost 2 years ago, so you can check it out here under this link. Next, I want to touch on the Neoantigen Meta-analysis that we performed. And we heard some background about neoantigens already in the previous presentation. I just want to briefly reiterate that. So cancers accumulate mutations in their DNA, so they can acquire their cancerous capabilities and grow and invade other tissues. And if a mutation is in a coding region, it can get translated into a mutated protein, and this protein eventually can get chopped up into different peptides. And in this context, we call them neopeptides. So these are mutated peptides. And if this neopeptide gets presented on MHC on the surface of the cancer cell, it can get recognized by T cells, which can trigger an immune response. So the T cell will notice that this mutated epitope is foreign, and then it will enable the killing of the cancer cell. In this context, we call it a neoepitope because it has been recognized by the immune system. And one, maybe I should mention this here, and one very important feature of neoepitopes is that they are highly tumor specific. So we talked about prostate antigens, for example, so they are shared across the, they're also available in healthy prostate tissue and also in the cancerous prostate tissue, but mutations are only present by definition. The somatic mutations are only present on the cancer cells, so the resulting epitopes are also very highly tumor specific. So theoretically, they make for very great targets. So this is also why the neoepitopes are so important, because we have some clinical relevance. Multiple studies have shown that the number of neoepitopes, for example, is associated with better outcome in patients receiving checkpoint blockade therapy, for example, anti-PD-1 or anti-CTLA-4. They showed that if you have more neoepitopes, the response is better. And the rationale behind that is that the T cells, they recognize these neoepitopes, but they have been silenced, so to say, by the cancer, by the inhibiting signals. And once they receive the checkpoint blockade therapy, they can finally break free of these inhibiting signals and then do the job and go and find the neoepitopes presented on the tumor cells and destroy them. We also showed in a recent study that spontaneous T cell responses against neoepitopes can be detected across a variety of different cancer types. There was no vaccination required, so the T cell responses were already there, and they can be harnessed to develop more effective therapies. And on the other hand, we have the computational challenge, because the predicting which of these mutations will give rise to neoepitopes that are actually recognized by T cells and can elicit an immune response, it still remains a major challenge. So there are many tools available, but they have so none of them are working perfectly right now. It's only a fraction of the neoepitopes can be predicted, and we still don't know why and what's going on. So this is exactly where our meta-analysis about these neoepitopes come in. Our rationale was to systematically analyze these experimentally validated neoepitopes that we have curated in CEDAR, and with the aim to better characterize their features and to ultimately contribute to improving predictions. Excuse me. So what makes our analysis unique is that this is the largest meta-analysis of actually experimentally validated neoepitopes. So there has been some previous studies, for example, but they mostly used predicted neoepitopes. For example, they went into some published sequencing data and they performed some predictions, and they did their analysis on these putative neoepitopes and not the epitopes that are actually experimentally validated. So as we've heard today, a lot of different experimental data is available in CEDAR, but for this meta-analysis, we focus specifically on human neoepitopes that result from single nucleotide variants in cancer patients. So we excluded, for example, frame shifts and fusion epitopes because there's only a few data points available compared to the single nucleotide variants that we are focusing on here. So we wanted to keep this data set as homogeneous as possible to get the best results. In total, we analyzed over 1,600 neoepitopes, 16,000 neopeptides from 180 published studies across 28 different cancer types. So this is a very comprehensive data set. And we heard about this before already, but just to reiterate here, it's important to remember that one peptide can appear in multiple different studies and publications and can be tested using different assays with potentially different outcomes. So, for example, I've highlighted this here. This example, for example, this peptide, for example, was tested in 3 different assays. 2 had negative outcomes and 1 had a positive outcome. And for our study, we considered this peptide to be a positive neoepitope when there was only 1 positive outcome. So if there's only negative outcomes, we don't call it neoepitope, we say these are the negatives. And even if there's only 1 positive outcome, that's enough to say it's a positive neoepitope. Okay, and following this, we checked all the epitopes and the assays that we have available, and 13% of peptides were validated as neoepitopes, so they had at least 1 assay that had a positive outcome, and the remaining 87% were considered negatives. So we began by looking at the top 100 proteins with the most tested neopeptides in CEDAR. So here, each column represents one of these top 10, top 100 proteins. And here we have the number of neopeptides and the number of neoepitopes in red and green. So the positives and the negatives are highlighted. And in the middle, we have each protein's length. And in the bottom panel, we show how frequently the gene is mutated in patients in CBioPortal. So we specifically choose these three metrics to visualize here side by side, so we can see if there's any correlations. The first, of course, as I mentioned before, the length of the protein is always interesting, because there's always the argument a longer protein can generate more epitopes and has a greater chance of producing neoepitopes thereby. But when we checked it, here we plotted the protein length versus the tested neopeptides in CEDAR. So there is, in fact, a significant correlation here, which makes sense because, yeah, as I said, the larger the protein, simply there's more sequence that can generate neopeptides. So we're distinguishing here again neopeptides, so just it's a mutation generated a mutated peptide. But then here we're looking actually at the peptides that were recognized by the immune system, so the neoepitopes. And as we can see here, the correlation is much weaker. So the correlation of actually tested positively tested neoepitopes to protein length is much weaker. So this shows while large proteins tend to produce more neopeptides overall, they are not necessarily more likely to be recognized. What we found instead is that driver genes are significantly enriched for neoepitopes, which we can see here. We tested the driver genes versus the non-driver genes, and we can see that the neoepitopes, were 21% of the neoepitopes and driver genes are actually recognized. So it suggests that the immune system may specifically recognize driver genes because these genes play a central role in tumor biology, and they are also under selective pressure. Because we saw that TP53 has the most neoepitopes, we wanted to investigate this protein specifically in detail. TP53 is very interesting in general because it is the most frequently mutated gene in human cancers. So it's also not surprising that it has the most neoepitopes in our database. In total, we found 152 unique neopeptides, and 35% of them were validated as neoepitopes. And interestingly, this is significantly higher than the global average that we saw of just 13% across all the proteins in the data set. So we have, again, here a visualization showing here in the top panel the length of the P50 protein, and these bars are the different peptides that are in CEDAR. So in red, we have, again, the negatives, the neopeptides, and in green, we have the neoepitopes that had a positive assay result. And in yellow, we highlight the mutation position. And as we can see here, the majority of the peptides are actually located in this P53 domain. So this is the P53 DNA binding domain specifically, and it plays a very important role for the TP53's function as a tumor suppressor. We also specifically see that many neopeptides arise from mutation hotspots. I indicated them by these vertical lines. So these mutation positions are known to be recurrent, and they're well known across different cancer types. So it also makes sense that we have many peptides generated by these mutations. So this brought up another question to see are the epitopes only studied if they are very often, if they occur in a lot of patients. So to better understand this, we compared the mutation frequency. Again, we looked it up in CBioPortal to see how often these mutations are actually mutated in different cancer patients. And we evaluated it with if there's a correlation against the PubMed IDs that are studying neoepitopes from these mutations. So here each dot represents a specific mutation, and we can see actually there's a strong positive correlation indicating that more frequently mutated, more frequently occurring mutations are also more frequently studied. However, we also identified this group, for example, here that are highlighted in blue. They do occur in a lot of patients, over 100 patients. So I would say they are recurrent mutations, but they are only studied in with one PubMed within one paper published in PubMed. So this points to a research gap. Because despite the high occurrence and the potentially clinical relevance, these mutations remain understudied. Yeah. So this again shows how CEDAR can be utilized to uncover research gaps and also to inform maybe some future studies. So next we looked at the neoepitope landscape across the different cancer types, and we investigated the fraction of validated neoepitopes versus the negative neopeptides. And so here we have the different cancer types, and then I'm going to walk you through the different panels. So here we just look at the different, at the number of peptides. So these are absolute numbers, and as expected, the highly mutated cancer types have a lot of tested neopeptides. For example, the top one is colorectal cancer, skin cancer, and lung cancer. These are very well known to be highly mutated cancers. But then here we looked at the percentage of, so okay, these are all the neopeptides that were tested, but how many of them, so the percentage of peptides were actually recognized as neoepitopes. And here we can see that colorectal cancer, for example, has a low neoepitope positivity rate. I think it was just above 6%. And then skin and lung cancer are significantly higher actually than colorectal cancer. This is actually an interesting finding because also lung and skin cancers are also known to be more responsive to immunotherapies when compared to colorectal cancer. So to account for the differences in mutational burden across the cancer types, we again wanted to look at the median mutational frequency derived from CEDAR portal and then normalize the number to account for this. So yeah, here we have the median CEDAR portal mutations per cancer type, and then here now we have a normalized normalized neoepitope fraction. And then we can see that after normalization, there are some important shifts. For example, colorectal cancer, so we compare now this panel and this panel. So this is the raw neoepitope fraction, and this is the neoepitope fraction normalized by mutational count. We can see that colorectal cancer is now much higher. It jumped up to 39%. So this might suggest that its initially low raw positivity rate was more a reflection of high mutational burden than a lack of T-cell recognition. So we could say, because colorectal cancer has so many mutations, the actual neopeptides and neoepitopes get diluted by this high number. Yeah, interestingly, a similar pattern was also seen in lung and lung cancer. We see that the bar gets much higher, but skin cancer did not move. And then we have down here some cancers like bile duct and pancreatic cancer. These are known to be low mutational cancers. As we can also see here, the bars are very low, but they actually have relatively higher normalized neoepitope positivity rates. So this might imply that in those cancers, when mutations do actually occur, they may be more likely to be recognized by T cells. Okay. So to conclude, we performed the largest meta-analysis to date of experimentally tested neopeptides and neoepitopes. We uncovered some patterns and uncovered some research gaps that can now inform the research community for hopefully follow-up investigations to have a more complete picture of these neoepitopes. So I only showed a few of the results that we have, and the full paper actually is currently under review. So again, keep an eye out for that paper and to review the full results. And this work was actually a very collaborative work, so I want to thank everyone that was involved. We have our group, and then we have Anna Carter from UCSD, who you just heard in the talk before, and we also work with Morten Nielsen from the Denmark Technical University. Yeah, again, I'm happy to take any answers, any questions in the QA box. Thank you.
Wonderful. Thank you so much, Zeynep, for going through all of that. Really quite cutting edge research, as you just heard. That paper is under review currently. So you're really hearing it here first, which is a really special aspect of these webinars and workshops that we can host across our resources. But I will give Zeynep just a couple of minutes to review the Q&A because there are a few questions for her before we wrap up for today. And another reminder, I did just drop the survey into the chat once again, with now less than 15 minutes to go. If you haven't already, we'd really appreciate your feedback on the general flow of the last 3 hours, the talks, any virtual or Zoom things you'd like to comment on, do let us know. But Zeynep, are you ready to take the first question?
Yeah. So the first question is CEDAR connected to CBioPortal and or other genomic databases? So I showed you that we are connected to quite a few resources using the Open Cravat collaboration that we had. But currently CBioPortal is not part of it, but it is planned to establish that connection specifically because of the utility that I presented here in this talk. So that's definitely on the to-do list to connect our data to CBioPortal to get these mutational frequencies. And also we want to provide so many papers. If they do these neoepitope studies, they start by sequencing data, and they have the genomic data deposited usually like in Geo or in DB Gap or other resources. So we want to include also those links so that users can go and also get access to the genomic data and perform some follow-up analysis. Okay.
Bjorn will be taking the next
I kind
question.
of jumped in. I wanted to. I think
Yeah.
I don't know thought this through. I don't want to want to give Zeynep some time to read stuff and answer one of the questions here
Thank
that
you.
I can. Why would you call a peptide positive that is only positive one time out of 100 tested? That is a really good question. And one of the things is like specifically in cancer and neoepitope research, that is not wrong. It is very possible, right, that if you have 100 different people, that only in one person something is positive and the others is not, because you have other issues surrounding it, like say the MHC alleles expressed in that person or so. So for us, essentially, it's like if it's never been found positive, there's no reason to call it an epitope. If it's found positive in one context, it is an epitope. Now, if you really want to know how positive it is, again, going to something like the Immunome browser where you have the summary over something that has tested multiple results or so, obviously you get more confidence the more something is reported to be positive in different people and results. But it's not like, even if it's just 1 out of 100, that doesn't mean it's not positive, because really we're not expecting responses to always be the same at all. Zeynep, go for it. I hope that gave you some time.
Yeah, thank you. So one question is, have you explored the function of immunogenic neoepitopes? Is this data accessible from CEDAR? So this is a really great question. So as I showed, I did like some high-level analysis comparing driver versus non-driver genes, but we did not go yet into like the detailed analysis of that. So actually the paper that we did on the Neoepitope Meta-analysis was already too long. So we might do a follow-up analysis with answering all these additional questions, because there's many open questions and it's a very interesting topic. So we could do something like looking at the GO annotations, looking at the different functional and at the location of the the cellular localization of the neoepitopes, for example. So there are many other different features to explore, but we haven't looked at it yet. But it's really a great question. Thank you. So this question is maybe related. What aspects, variables do you think need further investigation in the field of neoepitope discovery? As I said, there are many other features to explore. I think Hannah also has a paper looking at the subcellular localization of the epitopes that are actually recognized by T cells. So they found some very interesting findings there. Maybe Nina can also put that paper in our resources folder. Yeah. So let's see what we have.
I can do that. I'll keep updating the folder
Yeah.
with additional resources as they come.
Have you explored neoepitopes in mutation hotspots? Yeah, as I showed in the TP53 examples, we had several hotspots there. And I think it's also has been shown in general across the TCGA data sets that hotspots are very much studied. So there are also a lot of neoepitopes and neopeptides reported from hotspots. But I did not see a statistic actually if mutations from hotspots generate more neoepitopes. So that's also another interesting point to look into. So this question, I often use linear fold and Fornal toolbar structural template product via UNIF. Does your research use linear fold? Actually, don't know any of these tools. I don't know if anyone else can comment. But I'm not familiar with these tools, but we can take a note and look into it for sure. And same for adapt artificial soft tumor. I'm not sure if we have the fields to capture such tumors. Okay.
Good food for thought, things for us to take away and look at.
Definitely.
Thank you so much for those questions. Awesome. So
I think these were all the questions. Yeah.
Yeah, perfect. Thank you so much, Zeynep, for the wonderful presentation and for the questions, and Bjorn also for jumping in. We're basically at perfect timing. I will just close out the day with a few summary slides. So we're officially at the conclusion. Firstly, a big thank you to wonderful presenters, but also a bigger thank you to everyone who has stuck around and listened to all the presentations over the last 3 hours. We really hope you learned something and can take it away and apply to your own research. As a big reminder of where we started 3 hours ago, we are the Cancer Epitope Database and Analysis Resource. It is free and it was established in 2021 by the National Cancer, with funding from the National Cancer Institute, I should say. So we have two sides of the same coin, as everyone here has kind of mentioned. The database, which is depicted here, it catalogues experimentally derived cancer epitopes and the immunological context in which they're studied, so the assays and all of that information, the outcome. And you can access the CEDAR database at cedar.iedb.org. And really that's your entry point for everything CEDAR related. As I showed earlier, if you'd like to cite the resource, there is information there. Once our application program API is live, that will also be added under more CEDAR. So under here, all of that is where you should go to first. And the other side of the coin is the analysis resource. So these are all the tools, that aspects that Isabel and Hannah were talking about. So this is a web-based platform that provides integrated tools and pipelines for epitope prediction and analysis. and they can be accessed at nextgen-tools.iedb.org. And it's small here, but that little C denotes all of the CEDAR specific tools as well. So the resource is free, it is available, and we highly encourage you to take a look today. I did see in the poll that quite a few people hadn't accessed the resources, so I definitely hope today you've had a chance to learn more and take a look at it yourself. So a quick recap on the presentations. Right at the start, you heard from Dr. Alessandro Sette, the PI of CEDAR, giving an introduction from how it was created to how we track metrics and the general overview. Then you heard from Dr. Matthew Bussey, the senior curator, on how we actually curate the data and the overall database structure. After that, we honed in on how to query and export the results from the database. So you heard from Dr. Bjorn Peters, the co-PI of CEDAR, and then Dr. Zeynep Kosaloglu-Yalcin, our cancer bioinformatics instructor at the La Jolla Institute. And then we pivoted to talk about the tools. So you heard from Dr. Ibel Kari, our bioinformatics postdoc fellow, and Dr. Hannah Carter, our collaborator over at UCSD, on using the tools in a specific case study example. And then lastly, we heard from Zeynep once again on how all of that information that's been curated, the database, the tools, how that all can be applied to glean insights into what's available in terms of cancer research in the community, and what gaps, what can we learn from, and what can we do more to study and understand these diseases better. And with that, just a couple of reminders. You will receive a post-event email, and we will share the workshop recording with you. As Ibel mentioned, these videos will be posted to our YouTube, so you can go back and have a look. And the presentations are shared with you. They're in that Google Drive, but we'll be sure to share that again. And then one last mention of the survey. It is important, so please do, if you haven't already, open that up and share your thoughts with us. Once again, we'd love you to follow us on social media, on Blue Sky and X, and tag us in, something that you may have learned today or something that you might be using in your own research. And then, as we've mentioned, the team hopefully did a great job of answering your questions, but if there are any follow-ups, you can email us at cedar@lji.org, or if you would like to access our resources for non-academic purposes, you can email us at license.iedb.org for more information. And with that, thank you all once again for your time attending today's CEDAR short course. We hope you enjoyed it. And of course, we thank you for your interest in CEDAR and its resources. Thank you all, and we'll be in touch by email. All the best.